There are two parts to it. Both of them are quite important. You have your sequences uploaded to GenBank, and they will be there forever. Even you lose yours, you can always get back to it and download it. And also at the same time, like James mentioned, everyone else can make use of your sequences when it's up there. So first, we'll go through how to upload the raw reach data, and then the sequence service provider have trimmed the sequences and then supply to us and to you with these FASTQ files. So those are the ones that we're gonna upload in the first part. And in second part, we're gonna upload these genome assemblies that are made from the sequence reads. So let's go through the first part. It is uploading to SRA. I hope everybody has signed up to NCBI. If not, and I think please use the link in this presentation and then sign up according to what you prefer. Google ORC ID. We'll use this sequence read archive to upload our sequence reads. So it's run by GenBank, or a National Institute of Health in America. So these are all publicly available databases. So we'll use something called Sequence Submission Portal. If you click on the link, you will find this. And this is the submission portal at the GenBank or NCBI. The first thing you do is to click on what you are going to submit. So in our case, it is called SRA. When you click, you will be presented with this screen. So it would tell you what is involved to upload sequence to SRA. There are 5 steps. So you need to provide project name, project description, and then you have to describe your samples, and then upload sample metadata. And in the end, you upload your sequences. So these are the 5 parts that you have to go through. Click submit. And then when you do, you will be asked to log in. So that's what we're going to do. So every sequences you will upload, you will go through this login process. You can log in using different routes, either Google account or ORC ID, or the Microsoft if you have, or these NIH account or NCBI accounts if you have them. I suggest either Google account or ORC ID, because I have uploaded the sequence before. You will see that at the bottom part of the screen, so the sequences I submitted before. But the important point here is we have an option to do a new submission here. So that's where you should be clicking. So if you click new submission to upload your data, you will get this. Some of the fields will already be filled in for you. Please check them. Don't take it for granted that they will get it right. First, you provide your details, and then details of your institutions, and then your address, and then click on continue. And then you'll be asked to provide some general information, especially about your project or bioproject and your biosamples. So this is a new submission. So you will not have, you will not necessarily have this bioproject and biosample IDs that you had before. So that means you have to provide them and then select no and no. And then for the release date, we like you to click on release immediately following processing, so that you get your accession number as soon as possible, and then people can use it as soon as they have been processed. Later on, when you submit your genome assembly data, you would probably have received accession numbers for bioproject and biosamples for your raw reads that we're going to submit now. And you can use the same project and biosample accessions when we submit the genome assemblies. But for now, we'll say no and no. Okay, we'll do continue. And then you will notice that suddenly your 5 steps change into 8 steps. That's because we have to create a bioproject here, as well as a biosample here during this submission. So first, you have to describe what your project is and what the description of your project, and then give it a good project title. And then what is the relevance? It will ask you, is your project part of a larger initiative? Okay, so these data are at the bottom are also optional. So you can fill them or you don't, it's okay. And then press continue. So I got an error at the top. So this kind of errors, it's essential that you have to correct them. So in this case, the field, project title field needs to be filled in, and then project description also need to be filled in. So that's exactly what I did here. Project title, and then describe, why you are studying this bacteria or why it is important to have a genome sequence of this bacteria, the information that you would like to share with the public about your bacteria. And then I press continue. So in this case, the information was sufficient, and then you will be presented to enter the sample identification or description of your sample. So what we are submitting here is microbe. So I would like you to select this particular option here on this page. And then you will be presented with this option to submit the details of your sample. I suggest you use the built-in table editor. So that's where you click, and then you'll be presented with a table to fill in. So here, you need to give it a sample name, not the same as the sequence ID that you get from GetGenome, but you can use your own name. You can use your sample name here, and then give it a sample title. It's more descriptive. So there's a 1 star, a 2 stars, and 2 crosses, and a hash. So every asterisk column needs to be filled in. With the 2 asterisks, only one of them should be filled in. So in this case, there are 2, you only need to fill in 1, either strain name or isolate name. Once you fill in, click continue. And then now you will be asked to fill in a little bit about your raw reads and how they generate it. Library ID is where they make the sequencing library just before they sequence it. So that name needs to be unique. So normally I will just use your sample name and then add underscore LIB in capital letters for the library ID. You can give it a title for your sequences. So in my case, I just say WGS and then my sample name. And then library strategy is WGS, or WGS stand for Whole Genome Shotgun. So that's a sequencing strategy. And then library source will be genomic. We didn't do any library selection, so we'll select unspecified. And then library layout. So in our case, we'll only submit the paired reads, and then platform is the sequencing system that you use. So in our case, we use Illumina sequencing platform. And then instrument model, I selected here Illumina HiSec 2500, but in your case, it's Illumina NovaSec 6,000. So we'll just use whole genome shotgun or WGS. And then file type. So we are submitting FASTQ files. So select FASTQ. So this from library strategy to file type. So you all select from the little down arrow, of course, except the design description. That's where you just enter. And then you enter 2 file names, 1 and 2. And then make sure that this exact names that you are submitting. So please look at your sequence, raw sequence data. So that's number 1 and number 2. So under file name, submit number 1, and then file name 2, and then submit sequence number 2. So these should be paired end reads. Okay, so once you're happy with it, you can press continue. They will tell you to upload your sequences here. And I suggest you select web browser upload via HTTP. That means you are not using any other system except the web browser loading. You can drag and drop your files, or you can choose files, and then navigate to your sequence folder, and then select your sequences, and then click upload, and then it will take a while to upload to NCBI. But when it's finished, you will be presented with a summary of what you have entered, and please check exactly what you're uploading and make sure that the sequences are correct, sample names are correct, and then your affiliations are correct. And then all you have to do now is click submit. They will confirm your submission and then send you accession numbers for your bio project, bio samples, and your sequences within 2 or 3 weeks, depending on how busy they are. Be patient and you will get your sequences. The next step, uploading of the genome assemblies. This is slightly different from raw read upload. You will be uploading the contigs to GenBank. So we'll use the same link to SRA submission, and it's through sequence submission portal. So if you click on the link, you will get to the same place that we use for SRA sequence submission. But this time, you will select genome, click on submit at the bottom. You will be again asked to log in, and please log in the same way as you logged in to submit your SRA. And you will get to your personal submission profile page. And here you will see that I have submitted before some genomes that are processed and so on. So we'll click on new submission. We'll get to submission page. And what we are submitting is just single genome. Okay, we'll click continue. And you got this. And this involves 6 steps. So first, again, you have to describe who you are. So please make sure that the data that presented to you are correct. You can press continue if they are all correct. And then they will ask you some general information. So general information is quite long page. So divided into 3. So first, it will ask you if you already have bioproject accession and biosample accessions. And then this time, we will say yes and yes to biosample and bioproject section. And then enter the number that they will give you when you were submitting SRA project. And that we'll use the same project that you will receive for SRA submission, and then the same sample accessions you enter here. And then again, release date will release immediately. And next, the information about your genome assembly, date stamp from your sequence when it was assembled. And then assembly method is called spades. You have to give it an assembly name. Assembly name is simply your sample name and then dot and then 1 for assembly number 1, because you might have to modify it later. And then genome coverage and the sequencing technology, Illumina Novasec. Did your sample include the full genome? I say yes. We do have full genome, not just selected part of the genome. And then it will say, is it a final version? Yes. Is it de novo assembly? Yes. Is it an update of an existing submission? Say no. And then leave this box blank because we'll let it remove that they identify as contamination. Otherwise, you have to do it manually, and we don't want to do that. So let them remove automatically. Then you give a submission title. Like in my case, I write genome assembly of this species and this strain, and leave this box blank and a comment to NCBI staff. So that finishes the general info step. So the next step is the source of your sequences. Who can they contact? And then one important thing here is, do you want NCBI to annotate your genome sequence? I will say yes. And then click continue. So we are on the 4th step. Now you submit your files. Which of these 3 options describe this genome submission? I would say 1 or more chromosomes are still in multiple pieces because we are in a different context of the 1 genome. So we have to select number 2. And then how do you upload your sequences? Please select web browser upload via HTTP. If you click on that, you'll get a box here, and then you can drag your files. It's an assembly ends with FNA. So I'll show you next. So if you say continue, and then you can upload, or I changed it to .faster, but it's okay. It's called .fna in your annotation folder. So you should see the uploaded sequence here. Click on continue. There's an error in your sequence. So please read the error by clicking on like show report up here. They will tell you what's wrong with your sequence submission. And in my case, there are some problems with short sequences that are shorter than 200 nucleotides. So I had to open the sequence files in a text editor. We suggested Notepad++, and then find these sequences and delete them from the file, and then save your file as a different version. In my case, I saved it as a version 2, and then upload it again. You will get this. So that's a first version you uploaded, and then the second version I uploaded. And then you have to delete the one that is wrong. So we are nearly there. So once we got our sequences uploaded, and then press continue, and then it will ask you, what do you want to assign? Is it a sequence, a complete chromosome? We say no. And does belong to a plasmid, and no. And press continue. And now you can enter who are the authors of this submission. If you click onto the plus sign, you'll be able to enter more people here. Select unpublished. Reference title. just give the same title as your submission title. In my case, genome sequence of this bacteria species and this strain. If you're happy with this, and then click continue, you will be presented with a summary of what you have entered. So please check carefully. You just click submit and your submission is complete. And if they have any problems at that stage, you will be presented with the error messages. So please read the error messages and correct them if you can. So 2 things to submit. One is sequence reads to SRA, and then sequence assembly to GenBank.